So it's currently not doing that, but we'll record it and put it onto YouTube Live later. So thank you very much for starting early, very early. Hope you were able to do the quiz that's in the normal room. So any questions at all, just put them into the Q&A box. Remember, if you go to the student login, in Socrative.com, and it's available biologytutor.com, and you can do the quizzes as we go through this. And also, the next webinar is on Sunday, the 5th of March, and it's transporting plants. Again, if you found it useful, please recommend it to your friends, and we stream it live, hopefully, and record it on YouTube. We didn't stream this one live because it wouldn't. And we'll go through it again. Okay, let's plow through this. We're going to cover aerobic and anaerobic respiration, and hopefully by the end of it, you'll know more than you do now. Anybody's got any questions, just go ahead and ask them at any point. So what we're going to cover ATP and its importance. We're going to talk about glycolysis, which is how you split glucose. We're going to talk about the structure of mitochondria. We're going to talk about aerobic respiration, different respiratory substrates like fats and glycerol, and proteins, and the deamination of proteins to produce things that can get used in Krebs cycle. Okay, so first of all, where does the energy all come from? Well, it either comes from the sun or possibly through the oxidation of chemicals in the environment if you are a chemoautotroph. So most organisms, we get the energy from the sun, and well, you're thinking, well, I don't get the energy from the sun. Well, actually you do, because you end up eating autotrophs, or you eat things that eat autotrophs, because you are a heterotroph. So plants harvest solar energy, and then we convert that chemical energy into the products of photosynthesis. And they do that by having pigments that capture light and use that light energy to split water, and then use that to reduce carbon dioxide. So the products of photosynthesis provide the energy for the plants, and also the stuff that eats the plants, like us, and we are heterotrophs. Now, respiration is about taking the chemical energy of the organic matter, of the carbon-hydrogen bonds which have been produced by the plant, and then using those to produce the electrons, which can then be used to make the ATP. And we'll talk about that in some detail. So we start with light energy, which is used by autotrophs. Well, if it's light, then remember it's going to be photoautotrophs. These are going to trap the light energy and turn it into molecules that can be used by other things. So this is effectively carbon-hydrogen bonds, which can then be used to make amino acids, DNA, RNA, glucose, sucrose, everything in all living things, to be honest. Now that then gets respired by the autotrophs, and it also gets consumed by the heterotrophs. Now respiration then ends up producing ATP. ATP is used to carry out work, and also it releases heat because of there's inefficiency in some of these processes. So what do you need energy for? You need energy to create complexity, and energy to create order. You need energy to move things against their concentration gradient. You need energy to secrete substances, carry out endocytosis and exocytosis. You need energy to join monomers together into polymers, so replication of DNA and construction of proteins from amino acids. To carry out work, try not to avoid words about movement. So saying ATP is required for movement, you need to avoid that. But it is used for the contraction of muscles, because remember, it causes the change in orientation of the myosin head, which will help a sarcomere to contract. Anabolic reactions, so that's the making of big things from small things, like making proteins from amino acids, and maintaining body temperature. So we've got this amazing molecule, adenosine triphosphate. We're going to have a chat about that and the structure of it. And so why do you need ATP is if you made energy directly available to the cells, then there would be too much energy available in one glucose molecules. You'd need lots of energy, lots of enzymes in one single place to release the energy in that glucose molecule. And you'd need to hydrolyze lots of bonds. So instead, we've got something called adenosine, not adenine, adenosine triphosphate. Now, this is the intermediary for energy. Now, this is a concept that students do struggle with. When I mention it to them, I try to go with an analogy, which is that all of the things in your bedroom are all powered by electricity. And that electricity is generated from using many, many different sources of energy. So coal, gas, electric, nuclear, oil, wind, but it's all turned into a single currency, which is used then to power the resources that consume energy in your home. It's turned into a single common currency. So the source of the energy to power the objects in your house is not electricity. It's the fact that the coal, oil, gas, nuclear is used to create one common form, which is then used to power everything. So here's the structure of ATP. We've got an adenine, we've got a ribose, we've got 3 phosphate groups, and hence the T in the triphosphate. So it's a phosphorylated nucleotide, and it's used as the cell's universal energy currency, in that it's used by all living things to transfer energy for all reactions in all living things. And it's used to transfer energy from things that release energy to things that consume energy. So here's an ATP. We've got our adenine, we've got our ribose, and if we're just a normal nucleotide, we just have 1, so it would be mono, 2 is di, and 3 is tri. There you go, mono, di, and tri, and your adenine and your ribose forms your adenosine. Okay, different ways in which you can make ATP. We're going to talk about this in a little bit of depth. So I will just move on from that. Okay, moving on from that. I don't want to talk about that yet. Okay. There are 2 ways. Here we go. I'm trying to move on to the next one. There we go. That's the one. There are 2 ways in which you can generate ATP. One is through substrate level phosphorylation, and one is through chemiosmosis. Now, chemiosmosis is the way in which you generate ATP in mitochondria and in chloroplasts through the flow of electrons along a membrane to generate a hydrogen gradient. And those hydrogen ions then flow out through ATP synthase. So the way you generate ATP in both of those membranes is exactly the same way. Substrate level phosphorylation is the direct production of an ATP from taking a phosphate and joining it onto an ADP. And this is how you produce the ATPs in glycolysis and hence anaerobic respiration, and also in the Krebs cycle. Just move back to this point. So when we go from ATP to ADP, this is an exergonic, gonic means energy, exergonic reaction requiring ATPase. This is very different from ATP synthase. We're hydrolyzing, not breaking down. We're hydrolyzing the ATP into ADP, and we release the energy contained within the bond. We then use that energy to power things that require energy, so moving ions across membranes, endocytosis, exocytosis, etc. So we've got an exergonic reaction which is releasing energy. We've got an endogonic reaction, which is storing the energy. A point that's often missed by students, sadly, is that you've only got 5 grams of ATP in your entire body. And this is a generous teaspoonful or 2 sugar cubes worth. But you turn over some 80,000 grams, so 80 kilograms worth a day of ATP. So you're constantly interconverting ATP to ADP. It's constantly being used up from one form into another. Okay, substrate level phosphorylation is the direct transfer of a phosphate onto an ADP to form an ATP. And this occurs in the cytoplasm and also once per turn of the Krebs cycle. Now, chemiosmosis is movement of hydrogen ions across a membrane, and then those hydrogen ions flowing out through the amazing ATP synthase. So we could just take a moment and I will bring up on a whiteboard and we'll try and talk through this a bit more. So what you need for this process is you need a phospholipid bilayer and another phospholipid bilayer. Remember, the phospholipid bilayers are not permeable to hydrogen ions because the hydrogen ions are charged. And then we've got an amazing enzyme called ATP synthase, which does exactly what it says on the tin. It synthesizes ATP, and that synthesis of ATP is driven by a hydrogen ion concentration. And the hydrogen ions are flowing from inside the membrane, so in the gap between the 2 membranes, out into the mitochondrial matrix. Now, the pH of the intermembrane space, so this bit here is the intermembrane space, the pH is 1 pH lower than here, which is the mitochondrial matrix. So there are 10 times the amount of hydrogen ions here. And these hydrogen ions are flowing out through ATP synthase, and the ATP synthase rotates, and that generates ATP. Now, it's exactly the same in a chloroplast. In a chloroplast, this is the thylakoid membrane, this is the thylakoid interior, and this is the stroma. And we've got a concentration gradient of hydrogen ions. But the difference between the chemiosmosis in a chloroplast and chemiosmosis in a mitochondrion is how you achieve the ion gradient. So in a chloroplast, you achieve the ion gradient through the flow of electrons, which is powered by light. But in this instance, what we do is we have a chain of carriers. And these chain of carriers are embedded in the membrane. And we get an electron to flow down the chain of carriers. Now, as the electron goes down the chain of carriers, energy in that electron is used to move protons from where they want to be, which is in the matrix, to where they don't want to be, which is where there are more of them, which is inside the, between the 2 membranes. So the energy in the electron is used to do that, and the electron comes from a carrier blob, a big carrier blob called NAD. And it's called NADH because it's reduced, because it's carrying electrons, and that delivers electrons and becomes NAD again. Now the electron goes down a chain of carriers and gets to the last electron carrier, and at that point it joins with oxygen. With oxygen, hydrogens, and electrons, we form water. And this is what happens to all of the oxygens you breathe in. All of the oxygen you breathe in ends up becoming water. Now, as that's going on, the energy in the electron is used to move hydrogens across the membrane from where there are few hydrogens to where there are lots of hydrogens into the intermembrane space, and then those hydrogen ions flow out again. If we think, if I just clear this and we just compare it to what's going on in chloroplast, so we've got a thylakoid membrane, another thylakoid membrane, so we do a double membrane like this. Remember, in a chloroplast, you've got photosystem 1 and photosystem 2, and then you've got a chain of carriers, and then you've got electrons flowing down that chain of carriers from photosystem 2 to photosystem 1. The electron goes down the chain of carriers, powered by light, pushes protons across the membrane from the stroma into the intermembrane space, and then those electrons flow, not electrons, hydrogen ions, they then flow out, generating ATP. And in this instance, we take water and we split it to give you oxygen, hydrogen ions, and electrons, and the electrons are used to replace the electrons lost from photosystem 2, and those electrons end up, in the end, reducing NADP, another big carrier blob, to NADPH. So when you look at photophosphorylation, it's pretty much looks like oxidative phosphorylation backwards, because oxidative phosphorylation involves making water from oxygen, and photophosphorylation involves splitting water and the release of oxygen. Oxidative phosphorylation consumes a reduced carrier, and photophosphorylation produces a reduced carrier. Okay, now somebody had their hand up. If anybody has any questions that they'd like to ask now, then please do ask them in the Q&A button. No, nobody had their hand up? Okay. Very welcome to ask any questions. If I'm going too quickly, then do point it out. Okay, I'll move through this. Okay, let's go through glycolysis and Krebs cycle and link reaction, etc. So glycolysis, remember, occurs in the cytoplasm. So this is the first part, is we break down glucose into pyruvate. You take 6-carbon glucose and you break it down into 2 3-carbon pyruvates. Now this occurs in the cytoplasm. There's no carbon dioxide produced, there's no oxygen consumed. There's a small production of 2 ATPs. In fact, there's a production of 4 ATPs, but a consumption of 2 ATPs, and then there's also a production of 2 NADs, NADHs. Okay, if I take you through glycolysis on the whiteboard, so we start off with a 6-carbon glucose, and that gets phosphorylated twice. So we consume an ATP, get turned into an ADP, and we put on a phosphate, and we do it again to another ADP, and we put on a phosphate. Now, next step is that then splits, and that's called the glycolytic step. Lysis is to split, as in hydrolysis, and then that's going to produce 2 3-carbon compounds with a phosphate attached. So this is a 3-O's sugar, triose phosphate. Now that triose phosphate then turns into a pyruvate. So after the glycolytic step, everything happens twice because you've got 2 of them, because you've got 2 triose phosphates. Now this phosphate gets transferred onto an ADP, and that gets turned into an ATP. enough energy is released whilst you're doing that to take another phosphate and join it onto an ADP to form an ATP. And again, this happens on this side as well. So we make an ATP and we make another ATP. Now, these 4 ATPs are all being produced by substrate level phosphorylation, which is the direct addition of a phosphate to an ADP to form an ATP. Now, at the same time as this is happening, most importantly, is that there is also an NAD which gets reduced because it accepts 2 electrons and a hydrogen from the breakdown of the triose phosphate, and it gets reduced to form an NADH. So the triose phosphate is being oxidized, and we take the NAD, we take 2 NADs, and we turn them into 2 NADHs. Now, those are very important because they are then going to go into the mitochondria, and they're going to deliver the electrons to the electron transport chain, and that the electrons are going to flow down the electron transport chain, and the energy in those electrons is going to push hydrogen ions across the membrane, and then they're going to flow back into the matrix through ATP synthase. So what happens to these ones? These 2 NADHs that are produced in glycolysis in the cytoplasm is that they go into the mitochondrion, and they go through the outer membrane to the chain of carriers, and that regenerates the NAD because the electrons are dropped off. The electrons go down the chain and eventually join with oxygen to form water. Okay. So back to this. Now we go glucose to pyruvate, and then all of this occurs in the mitochondrial matrix. So you then have link reaction and the Krebs cycle, which occur in the matrix of the mitochondrion. So running through glycolysis step by step, we start off with glucose, we phosphorylate it, then we phosphorylate it again to form fructose bisphosphate. Then it splits in the glycolytic step to form 2 triose phosphates. Then we phosphorylate 2 ATPs twice, which gives us our net gain of 4 minus 2, which equals 2 ATPs. And then we make 2 reduced NADs because we have produced the electrons which are enough to reduce the NADs. Okay, we go into a mitochondria. So mitochondria, remember, were originally bacteria incorporated into larger cells, which then became eukaryotes. But they have a single circular loop of DNA. You got all of your mitochondria from your mum because they were in the egg that made you. Most importantly, they've got an outer membrane and an inner membrane. And remember, the gap between the membranes is where the hydrogen gradient builds up. You've got cristae, which are infoldings of the membrane, which are there to increase the surface area so you can have more surface area for oxidative phosphorylation to occur on it. You've got ribosomes, so that protein synthesis can occur and you can make the enzymes required for the link reaction and the Krebs cycle. Okay, so mitochondria, they've all got an inner and an outer membrane, and we form cristae. There's a gap between the inner and the outer, which is known as the intermembrane space, and then you've got a mitochondrial matrix. Little ribosomes. Now keep moving through this. Okay, so intermembrane space, which is the gap, matrix, which is where the enzymes of the link reaction, the Krebs cycle are, and then we've got cristae, which are infoldings. large infoldings, increasing the surface area. And don't put just increasing the surface area, make sure you include increasing the surface area for reactions to occur on those membranes. And then with the ATP being generated by ATP synthase, which is on those membranes. Okay, so things that are on the inner mitochondrial membrane, we have the electron carriers, and these electron carriers are going to accept the electrons from NADH and also from FAD, which we're going to talk about in a minute. And those carriers are on the inner membrane. At the end of the electron, they join with oxygen to produce water. I know I'm going on about this a lot, but it is important. The energy in those electrons moves the hydrogens across the membrane, and then the hydrogen ions flow out again. Okay, so we had glycolysis, which involved going from a 6-carbon compound to 2 3 carbons because it's split in the glycolytic step. We produced 2 ATPs through substrate level phosphorylation. Well, we produced 4, but we've consumed 2, so there's a net gain of 2. We've produced 2 NADHs, and we've produced 2 pyruvic acids. So what happens next is we have the link reaction. And the link reaction occurs, remember, inside the mitochondrial matrix. So we've got an inner membrane, the outer membrane, and we'd have pyruvate. Now, the pyruvate gets decarboxylated by a decarboxylase enzyme, which is an enzyme that removes a carboxyl group. Now, do remember that there is oxygen here. There is oxygen there and there, but the oxygen in the carbon dioxide has nothing whatsoever to do with the oxygen you breathe in. The oxygen you breathe in becomes water, and the carbon dioxide is produced by the decarboxylation of carbohydrates, fats, and proteins. So we have a decarboxylation by the decarboxylase enzyme. At the same time, there is a dehydrogenation by dehydrogenase enzymes, which produce 2 hydrogens, and these are then picked up by NAD, which gets reduced to form an NADH. NADH goes off the electron transport chain, delivers the electrons, and therefore becomes oxidized. It goes back to becoming NAD. You've got a chain of carriers. The electrons that are being delivered then go through the chain of carriers, join with oxygen to become water, pushes protons through against their concentration gradient. They flow out again, forming ATP. Now we've decarboxylated the pyruvate and we've lost the carbon dioxide, so we're now left with a 2-carbon compound, which is acetyl, and that joins the carrier blob. In fact, I think it's the pyruvate that joins the carrier blob to start with, but we form acetyl coenzyme A, and that leads us then on to the Krebs cycle. So this step from pyruvate to acetyl CoA is the link reaction, which produces carbon dioxide and NADH. Doesn't produce any ATPs directly. Remember that 2 pyruvates are produced for each glucose, so that the link reaction occurs twice per glucose. And then we go on to the Krebs cycle. So now we've got a 2-carbon acetyl CoA. Just ignore the coenzyme A, it just regenerates and goes round again. And that's going to join with a 4-carbon oxaloacetate to form a 6-carbon compound. 6-carbon compound gets decarboxylated by a decarboxylase enzyme, gets dehydrogenated, which joins with an NAD to form an NADH, which goes off to the inner mitochondrial membrane and delivers electrons and drives the protons across the membrane, all that sort of stuff. Produce a 5-carbon compound. That 5-carbon compound then gets turned into a 4-carbon compound. Again, we produce 2 hydrogens, which then get reduced by another NAD, forming an NADH, which goes off to the inner mitochondrial membrane. Electrons flow down, join with oxygen to produce water, push the protons across against the concentration gradient, and so on. Then we've got a step with a 4-carbon going to form another 4-carbon. There's some oddness that occurs here, which is that firstly, you form an ATP. Well, it's not, it's a GTP, but it becomes an ATP. So most textbooks call it an ATP. You form an ATP through substrate level phosphorylation. This is the direct addition of a phosphate to an ADP to form an ATP. So one of these is produced per turn of the Krebs cycle, therefore 2 are produced per glucose. Now, at the same time, you release 2 hydrogens, and these are of slightly lower energy level, so these are picked up by a different carrier, so not NAD, they're picked up by FAD, and it gets reduced to form an FADH. Now, that 4-carbon compound then gets oxidized again to release another 2 hydrogens, and these get picked up by another NAD to form another NADH, which again goes off to the electron transport chain, and then we end up producing, back where we came from, another oxaloacetate. Now, the only things you need to know in link reaction and Krebs cycle, the only things you need to know the name of are the acetal, or starts off with acetal CoA, the oxaloacetate, and the citrate. The 5-carbon, the 4-carbon, the intermediates, you don't need to know. What you do need to know is the big picture, is that what you have here is a progressive decarboxylation and a progressive oxidation of the citrate through a series of steps to oxaloacetate, and that you're removing the electrons and the hydrogens, and you're using those to reduce the NAD to NADH, and the FAD to FADH, and the NAD to NADH, and that you end up producing 3 NADHs and 1 FAD, and also you produce 1 ATP through substrate level phosphorylation. Okay, I'll just clear and stop that. Let's go back. Okay, here we are. So we're back here with glycolysis going to 2,3 carbons, then into the link reaction, which, if you remember, occurs in the mitochondrial matrix. So we take a pyruvate and we decarboxylate it, and we take 2 hydrogens and dehydrogenate. Produce the acetyl CoA, joins with a 4-carbon to produce a 6-carbon, progressive decarboxylation, and dehydrogenation, producing the reduced carriers. So glycolysis, link reaction, Krebs cycle, and then the electron transport chain to take all of the reduced carriers that you've made, so the NAD and the FAD, and then end up producing the ATPs as the punchline. Remember, glycolysis occurs in the cytoplasm, doesn't consume any oxygen, doesn't produce any carbon dioxide. Link reaction and Krebs cycle, these occur in the matrix of the mitochondria. Okay, link reaction, pyruvate joins with coenzyme A, forms acetyl CoA when you decarboxylate it, remove the carbon dioxide, and then you reduce the NAD. Krebs cycle in more detail than me just scribbling away, in fact, the same amount of detail. 4-carbon compound joins with acetyl 2 carbon to produce a 6-carbon, decarboxylate, dehydrogenate, 5-carbon, decarboxylate, dehydrogenate, formation of an ATP, formation of an FAD, which is a lower energy carrier. So it's going to make slightly fewer ATPs than the NADH. Produce another NADH, again through oxidizing to here to form the oxaloacetate, and then the process continues round. Remember, this occurs twice per glucose because you produce 2 pyruvates. And then we go to the final stage, which is the oxidative phosphorylation and then the use of those hydrogen ions. So we've got the NADH, and then that's used to regenerate back to the NAD, produces the electrons. The electrons go through NADH reductase, cytochrome reductase, cytochrome oxidase, eventually join with oxygen to produce water. Now remember, whenever asked about oxygen, you've got to repeatedly say that it's the final electron acceptor in the electron transport chain. Very important. The chain of carriers that are on the inner mitochondrial membrane, and remember there'll be another membrane out here, and this would be the matrix, and this would be the intermembrane space. The hydrogens are being moved against a concentration gradient. In fact, there are 10 times as many hydrogens in here. At the end of it, the hydrogens flow out, making ATP. Talked about that, talked about that, talked about that. I haven't talked about that so much, but a point to make is that, remember, the ATP synthase physically rotates. It actually goes round. If anybody's interested in seeing this, there's a fantastic animation at www.johnKyrk.com. Not quite sure how you pronounce it, but if you go there and have a look at it, johnKyrk.com, there's a superb animation which shows the ATP synthase molecule rotating. Just a point to make about the NAD and the FAD. So the NAD is going to push 1, 2, 3 hydrogen ions through the membrane, whereas an FAD is only going to push 1, 2 hydrogen ions through the membrane. So one NADH ends up producing approximately 3 ATPs, and one FADH ends up producing theoretically 2 ATPs. I say theoretically and approximately because some hydrogen ions leak back across the membrane without going through ATP synthase, so there is a loss of some of the hydrogen ions, so they're not used to make the ATP. Okay, any questions? Now is the time to ask them. There'll be a second Socrative quiz that I will put up now. It's at socrative.com, A-level biology tutor in the student login. I will just make sure that comes up now. Okay, so there's another quiz, so you can head over there and do that. Any questions? Now is a good time to ask them in the Q&A box. If you haven't got any questions, then please just do the quiz. Well done, lots of correct answers coming up in the quiz. Well done. Anybody who hasn't entered, then remember, it's go to socrative.com, put A-level biology tutor in and it should be running automatically. Any questions, people? Lots of you streaking ahead. One of you's finished the quiz already. Well done. 100%. Excellent work. There's another quiz at the end, which is a slightly longer one. Okay, I think most of you have finished there. Just check. Almost finished. Okay, a few more points to talk you through. And remember, any questions, anytime you want to ask questions, then please do ask them. So how many molecules of ATP are produced each of the 4 stages? This is quite a common thing to look at. Remember that each molecule of reduced NAD is going to produce 3 ATPs. Each molecule of reduced FAD produces 2. And remember, there is the glycolytic step. So the glucose splits into producing 2 3-carbon triphosphate. So everything that happens after that happens twice. So glycolysis is going to produce 2 NADHs. It's going to produce no FADs. It's going to produce 2 ATPs through substrate level phosphorylation, which is obviously 4 minus 2, because you produce 4, but you consume 2 in phosphorylating the glucose to form a glucose biphosphate. These 2 NADHs, if there is oxygen present, then those NADHs can go to the electron transport chain. If there's not oxygen present, then they're used to produce either lactic acid or ethanol in anaerobic respiration, which we're going to talk about in a minute. So if those NADHs can be consumed in oxidative phosphorylation, then they are used to make 6 ATPs, because there are 2 NADHs and you make 3 ATPs per NADH. So in total, if there is oxygen present, then the number of ATP molecules produced from glycolysis will be 8. However, if there is no oxygen present, then the number produced will be 2, because the 2 are being produced through substrate level phosphorylation, which doesn't need oxygen. Okay, link reaction produces 1 NADH, but it happens twice per glucose, so it ends up producing 2. If there's oxygen present, these 2 NADHs go and produce 2 times 3, so 6 ATP molecules. Krebs cycle, we're going to produce 3 NADs, and we're going to do it twice, because there's 2 turns of the Krebs cycle for every glucose. So this ends up producing 6 NADHs. We're going to produce 1 FAD, but we're going to do it twice. And we're going to produce 1 ATP from substrate level phosphorylation, but we're going to do it twice. So that ends up producing 18 ATPs from the NADH, 2 times 2, which gives you obviously 4, which gives you the other 22 ATPs from oxidative phosphorylation, which in total gives us some 38 ATPs per glucose molecule. Now, the total of 38 ATP molecules per glucose is theoretical, and generally this doesn't happen. And why there is the energy losses is that ATP is used to move the pyruvate, the reduced NAD, and the reduced FAD across the mitochondrial membrane. So there is an energetic cost to getting it into the mitochondria. Hydrogen ions will then, some hydrogen ions will leak across down their concentration gradient back into the matrix without going through ATP synthase. So there'll be a loss there. So on average, there are about 30 to 32 molecules of ATP produced per glucose. Okay, on to anaerobic respiration. Now, anaerobic respiration is nothing special. Anaerobic respiration is just glycolysis. That's it. It's just glycolysis. And it's glycolysis and then doing something with the NADHs to regenerate the NADs so that glycolysis can continue. There's nothing tremendously clever here. Now, there's two forms of regenerating NADs. Either you use the NADHs to reduce ethanal to ethanol, or you use the NADHs to reduce pyruvate to produce lactic acid. So anaerobic respiration is respiration where you don't need any oxygen. There's two types. There's lactic acid and alcohol fermentation. Lactic acid is done by us and bacteria. Alcohol fermentation is done by yeast and plants. So here's lactic acid fermentation. You take the glucose, you carry out normal glycolysis, and you produce NADH. So let's just talk about that with the whiteboard. So remember, you've got glucose, you phosphorylate it with the 2 ATPs to produce the 2 ADPs. That then splits. We've got 2 molecules of triose phosphate. Remember, that produces 4 ATPs through substrate level phosphorylation, where you take 4 ADPs and you phosphorylate them to produce your 4 ATPs. This gives us a net gain of 2 ATPs because you consume 2 and you produce 4. At the same time, this is where you reduce an NAD to an NADH. Now, conventionally, the NADH is going to go off to the electron transport chain, and it's going to be regenerated by donating the electrons. But under situations where your expenditure of ATP exceeds your abilities to make that ATP through oxidative phosphorylation, so you can't get enough oxygen into your mitochondria, so you're sprinting for a bus or you're running away from a tiger, either of those things, the only way you can continue to make ATP in excess of your ability to make it aerobically is to make it anaerobically. And in order for anaerobic respiration to continue, you need a supply of glucose and you need a supply of NAD. Now that's the problem, is how do you have the supply of NAD? And the solution is to take the NADH and use the electrons to reduce the pyruvate to produce lactic acid. So that means you can keep regenerating the NADs, and that this process can continue on making a net gain of 2 ATPs. You keep feeding in the glucoses, and then you keep using the NADHs to reduce the pyruvates to lactic acid. Now, what happens in plants is a very similar process, and plants and yeast, first of all, they take the pyruvate and they decarboxylate it to produce ethanol. And that then gets reduced to form ethanol. And again, this process is, the purpose of this process is to regenerate the NADs so that glycolysis can continue with the net production of 2 ATPs. So again, anaerobic respiration is nothing clever. It is just glycolysis with something else happening. Okay. Cardo, what's your question? Ask a question, Cardo. You can put one in the Q&A box. Excellent. How is the liver involved in aerobic respiration? The liver is involved in aerobic respiration to anaerobic respiration. I think you're talking about lactic acid and the regeneration of pyruvate, which can occur in the liver. Okay, I will. Okay, let's keep moving on. So that was anaerobic respiration in yeast and plants, which I talked about, and then other respiratory substrates. So there is a big focus by students on glucose. Remember, glucose is only one of the things which can power you. The other things that can power you are fats, which can be hydrolyzed into fatty acids and glycerol, and then also you can be powered by the breakdown of amino acids and deamination of the amino acids to produce an organic acid, which can then power Krebs cycle. So do remember that there are other respiratory substrates that you can use to power respiration. So carbohydrates are important, and glucose is particularly important because there are some cells which can only use glucose. But most other cells can respire lipids and proteins. So you can take lipids and you hydrolyze the ester bond to produce the fatty acids and the glycerol. Remember, the glycerol is the 3-carbon compound to which you have the ester bonds, and then you've got the fatty acids coming away and all this sort of stuff. And what you do is you hydrolyze the ester bond and you take your 3-carbon glycerol, and then the 3-carbon glycerol gets phosphorylated and turned into triose phosphate, which can then enter glycolysis and start the process. The fatty acids, remember the fatty acids range in length. Those fatty acids are then hydrolyzed into 2 carbon fragments, and those 2 carbon fragments go into the Krebs cycle. So what we have is, I'll talk about amino acids in a minute. The lipids are hydrolyzed to give you glycerol, and the glycerol is then phosphorylated to form triose phosphate, and the triose phosphate then produces the NADH and then goes into the link reaction in the Krebs cycle. Fatty acids are hydrolyzed to produce 2 carbon fragments. That 2 carbon fragment then enters the Krebs cycle. And it depends on the nature of the R group as to where the amino acids enter. If I just show you on the whiteboard. So remember, if you've got an excess of amino acids, then you're going to take the amino acid and it's going to be deaminated, so removal of the amine group, and that's going to be used to make ammonia then quickly into urea. You're left with a 2-carbon compound, and that will then enter the link reaction. Well, sorry, enter the Krebs cycle because you've made a 2-carbon compound. Now, it depends on what the R group is, because if the R group is something more complicated, so you've got more carbons here in the R group, then it's going to go in at a different stage of the Krebs cycle, because you're going to make a bigger organic acid. So when you see the products of amino acid breakdown entering at different stages, then that's because the R group is going to change what the organic acid is after it's been deaminated. Right, final quiz to get running. And then any questions, please feel free to ask them. I'll just get the final quiz running. So I'll just finish that. Okay, the final quiz is running. Some of the questions are the same, some of them are different. Hopefully it won't take you too long. Any questions, then feel free to ask them. I'll be happy to answer any if I can. Any questions at all, people? Anything you'd like me to go over again, then just mention it now. Okay, I'm going to finish the YouTube live bit, but I'll keep talking to you in a moment. So I'll just log off from that. The next webinar is on the 5th of March, transporting plants. If you found it useful, then tell your friends or subscribe on Twitter. I'll just stop the live stream. Okay. Any questions, people? Yeah, some of you applying through the quiz, well done. Good work.